It started.
I know. Okay, so this is the organic chemistry lab. And what we would ordinarily do in class is, before I ever walked you through anything, people would be forming groups. And I would always tell them, your group has two options. You got a whole bunch of tests to do. You can divide the tests up amongst the different people. You get done faster, but the people that are not doing this test, the people that aren't doing this test don't have any experience doing that test. The people that do this test and screw it up, everybody winds up having bad data on their labs. So there's an upside and a downside. For the other groups, they can do everything. But I always tell them, you have to really pay attention when I walk you through the procedures, because you have to be pretty organized to be able to do everything in the time it takes to do a regular lab. Now, I'm gonna be your lab partner. We are going to do everything, as in, I am going to do everything. Hopefully, it won't take a super, super long time. After all, I kind of know how this works. I kind of know how the directions are. But we're gonna go step by step by step, sort of. Is that before we start, you probably want to have the sheets for this lab somewhere where you can see them, because you're gonna be writing stuff down on them according to the results that we get. Now, right off the bat, this is the most confusing thing about part one. We're in a lab. We're about to run a bunch of tests. We would like to know if the tests are actually working the way that they're supposed to be working. And that's what part one is. We run tests on things where we already know what's in them. We already know what tests we're running. So every time we run a test, we need to know what it's supposed to do, or we won't know if it's not working. So when you look at the first part one tables, the first column is always predictions. If you're running a sugar test on something with sugar in it, do you think it's going to give you a positive? Here's the thing. We have, oops, oh crap. I already started out as a typical lab partner. I spilled stuff on the table. And I also found out that when I set this up, I grabbed what I thought was sugar solution, and actually it was starch solution. So I have 2 bottles of starch solution. I have a bottle with starch in it. That is a complex carbohydrate. We have a test that will pick that up. It won't pick up any of the other substances. We have a bottle with a sugar solution in it. That's a simple carbohydrate. We have a test that will pick up sugars. It won't react with starches. It won't react with any of the other stuff. We have albumin, which is egg white protein. It's a protein. It's not a carbohydrate, it's not a lipid, it's a protein. We have a test for proteins, and we also have a test for the bits and pieces of proteins, amino acids, is that it shouldn't react with the other stuff. We have vegetable oil, that is a lipid, not a carbohydrate, not a protein. We have a test for lipids, it shouldn't react with anything but lipids. You see where I'm starting to go? We have water. Water is our bottom line comparison. It's got nothing in it but water. And so none of the tests for any of these other substances, you wouldn't expect to react with water. And that's the thing. If I run a protein test on the sugar and I get a positive, I know my sugar is messed up or my test is messed up. And before I run my experimental tests, I need to figure out what's wrong and fix it. That's what we're doing for part one. We're running basic tests. Now, in order to be organized properly, there's some parts of some tests where you have to set them up and set them aside for a while, and that's what we're gonna do to start. So I'm gonna skip around on the tests, but you have all these directions on the sheets. First thing I did was I set up my hot water boiling. I keep turning this down. I don't want it super, super boiling because I don't want it to all boil away, but I need it at 100 degrees Celsius so that it's a reliable temperature for my tests. My first test that I'm going to start is a basic lipid test. Lipids are fats and oils. If you've ever gotten, French fries in a bag, you know that if they sit there for a while, the oil from the French fries will soak into the bag. And unlike water, it's not gonna evaporate out. It'll just stay there. It'll leave a wet spot. That's what we're looking for in the lipid test. So we're using basic paper towel. Starch settles, so I have to stir it up. Now, I should be doing this. Your lipid is here, second page, grease spot test. I'll do this in the order that it's in here. I'm gonna take water, just make a little spot. I'm gonna take starch solution, just make a little spot. Now I could have labeled the sheet, but since I'm going in order from left to right, I know what I'm gonna have there. I have sugar solution. Let's stir that up just in case. It shouldn't need it. I'm gonna make a little spot. I have vegetable oil. This is my lipid. I'm gonna make a little spot. If I can get it to go into the dropper. And I have protein solution, egg albumin, dissolved in water. I'm gonna make a little spot. Now, in order to run this test, I have to give this stuff a chance to dry. I have to give it a chance to evaporate. The lipid won't evaporate. So I'm gonna set this aside and give it a chance to go. Now, some of the stuff soaked through to the table, so always wanna eliminate any contaminants you have that you know of. We have a test that's gonna run for amino acids. It's the ninhydrin test. And I just need to make a little mark. It also is gonna use drops on the paper, and the drops have gotta dry up. It's gotta leave just what's dissolved in the drop on the paper. If there's still water on the paper, when you put the reagent, the test substance on it, it will spread out through the water drop and it'll get too thin, you won't get a reaction. So, again, a little drop of water. I'm gonna go clockwise so I don't need to mark everything else down. Little drop of starch. Little drop of sugar solution. What's next? Little drop of vegetable oil. Little drop of protein solution, albumin. That also has to have a chance to evaporate, so that goes off to the side. Now we're gonna run the first test tube test. That's the Benedict's test. It's on the front of your sheet. We have, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We need 10 test tubes. Okay. Never trust the person that used the test tubes ahead of you. So I'm just gonna rinse them out really fast. With what goes on in the test tubes, a good rinse should be enough, as long as there's no trapped food. A little bit of extra water in the test tubes is not gonna interfere with the tests. So fill it up, shake it out. 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 Fill it up. This is going to be a sugar test, and it is the only test that needs to be heated in order to work. What happens sometimes with these lab groups is I look across the room and they've got other tests in the boiling water. And you know something? They won't work if you put them in boiling water. This one's the only one that will work in boiling water. If you're looking at your table, we're gonna set 2 test tubes up with water. Again, I'm gonna work from left to right. So 2 test tubes are gonna get 6 drops of water. Let's see how well I can count. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 2 with the starch solution. I'm gonna stir it up again just to be sure. Starch is a really big molecule. It doesn't dissolve well, it settles. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Sugar solution into the 3rd set. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Vegetable oil into the 4th set. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And protein solution, albumin, in the last one. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Now the front row is going to get the chemical that actually tells you whether there's sugar in there. The problem we wind up with is the results rely on color changes. And for Benedict's, there's actually a few different colors that will tell you that there's sugar present. The Benedict's itself is blue. If you get a weak color change, it turns green. If you get a little bit stronger color change, it turns yellow to orange. A strong color change, it turns red. A really strong color change, it gets coppery colored. This blue is actually oxidized copper. And heating it up and reacting it with sugars actually converts that to metallic copper, and that changes the color of the blue. Because it's a color change, when you run tests with things that have color, you can't always be sure whether your results are because you got a positive or because you just mixed 2 colors together. So we run what are called color controls. So the first groups are going to get 6 drops of Benedict's. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And the next set are going to get my blue water. I've mixed this with food coloring to make it match the color of Benedict's. So any change we get from this is purely a color change. It's not a chemical change because there's no reacting chemicals in here. So this goes into the back ones. This is our color control. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Okay. These have got to go into the boiling water for 3 minutes. I'm not putting them all in because I can't keep track of them if I put them all in. What I'm going to do is put my 2 waters on the left side and my 2 starch solutions on the right side. Set them up so that they'll stay that way. And okay, now I've got to let that run for 3 minutes. The second set of tests is going to be the Lugol's test. I'm going to grab myself another test tube rack. I have to swap out my other glasses so I can see across the room and see the clock. I know how I started, but I'm not getting a clear look at it. Okay, we're coming around on 1 minute. 1, 2, Okay, we're not quite to a minute and a half, so I've got time to wash these. Okay, it's coming around the 2-minute mark. I just realized, this is a good thing about having this on video, is that you needed to tell me what the starting colors were. Now the thing is, all of this stuff starts as blue. Sometimes the vegetable oil, because it's yellow, will get a little bit green. This one doesn't seem to be even doing that. So our starting color for all of these is blue. This is the only one where you actually mix stuff together and record the colors as the starting color, because this is the only one that doesn't react on contact. It has to be cooked first. And we're at 3 minutes, and I just realized that Joan hasn't found me my test tube tongs yet, so I'm gonna break a rule. You should not do this. These are Pyrex. I can trust the tops to not be that hot. So I'm gonna take them out by hand. So, for the water, still blue. For the starch solution, still blue. Now we're gonna put the sugar solution in and the vegetable oil in. And I'm gonna set up 6 more test tubes for our last test tube run. This is why even if you do things as a group, everybody gets stuff to do, because there's test tubes to set up and things to record, all sorts of fun stuff. I always tell people that miss this lab, yeah, you can come in and make it up, but it's not an easy thing to do, and it takes you a long time. Okay, coming around on 1 minute. Now, depending on how long this takes, I may do part 1 as a single video or as 2 videos. Once we get this, the results on this, I'll figure out how far in we are and then decide whether to do a second video. Okay, coming around on 2. I don't know if you can see, we've got a change going on down here. Whenever you run this test, you have to be ready at the end of 3 minutes to get your stuff out, because a hot test tube is going to keep cooking. So here's our 2 sugars. Here's our vegetable oils. I'm going to put the 2 proteins in. So there's the sugar control. There's the sugar test. Here's the vegetable oil control. It's kind of changed a bit because we cooked it, but it hasn't turned green or yellow or orange or red, so it's not a positive. And the color change for just the colored water didn't change at all. So we're kind of waiting on this. Here's our lipid test. It's already dried out. So I'm gonna let this hang so I can point at it. Here's the water, here's the starch, here's the sugar, here's the vegetable oil. You see the light come through it? And here's the protein. That light coming through the paper is a positive because the lipid hasn't evaporated. Everything else left a little bit of spot, but it hasn't left a wet spot. And that's what you're looking for in the lipid test, whether it leaves a persistent wet spot. Okay, about 30 seconds to 3 minutes on this one. Once we take this reading, we will stop this and do a video for the rest of part 1. I try as much as I can not to go over 25 minutes on a video. And there we go. Now this is the protein, and notice it did change color. It's kind of a weird blue, not the blue that it started as, but that's not a Benedict's positive. And that's the thing, just any old color change isn't what you're looking for. It's a particular color change. And in this case, it didn't turn green or orange or red or copper, and the color one didn't change at all. So something happened in here, but I'm not really sure what. But it's definitely not a positive for sugar. So we've done the results for the Benedict's test, and you should have those recorded on your sheet. And we have the results for your lipid test, the grease spot test, and those should be recorded on your sheet. And we're going to do one more thing. I'm just going to tell you this. I'm going to take a drop of, well, I'm going to do this off camera because it isn't actually on my cart. Take a drop of ninhydrin, which will react with amino acids, but only if you hit the combination with light. I'm going to put a drop of ninhydrin in the middle of each of these spots, and then I'm going to stick it under a light for a while. And if it's a positive for amino acids, it should turn purple. Ninhydrin is very antsy. Sometimes it works and sometimes it doesn't. But we are not going to do part 2 tests with this. So this is really just a demonstration of another way the chemistry works, where sometimes energy like light will get in and mess with the molecules to make them do what you want them to do. So we're going to stop the video there.
Sorry, it doesn't want to stop.
Well, we're going to try to stop the video there.